For next lesson, we have the peptide bonds, which are basically the bonds that attach one amino acid to another. Now, the peptide bond is a covalent bond, which I think I've already mentioned this one when we have the review video on the chemical bondings and specifics. So a chemical bond is a chemical bond, specifically a covalent chemical bond formed between two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, causing the release of the water molecule. So going back to the structure of amino acid, we have the central carbon atom, and of course we have the side chain, and of course one hydrogen atom attached to it. And then we all know that it has an amino group attached to it and a carboxyl group attached to the alpha carbon. Now, if this is one amino acid, we should also have another amino acid. Right? So COO2. All right. Now, in this example, we already have two amino acids. And again, a peptide bond is what attaches one amino acid to another. Most especially, it attaches the carboxyl group. It attaches the carboxyl group of one amino acid to the amino group of the other amino acid. So let's see what will happen. So in this reaction, we usually remove OH, or the OH from the carboxyl group. So if we remove that one, we will have the OH here. And then one hydrogen atom will be released from the amino group. So if this specific hydrogen atom will be released, we will have the H here. So OH reacting with H, we will have H2O, which is a water molecule. So if we remove the OH here and the H here, what will form is CO here and then the NH here, right? And so the attachment between the CO and the NH will be known as your peptide bond. Right. So this one is your peptide bond. Now, the dehydration synthesis reaction is the same reaction that you observed in my previous example. So the dehydration reaction is also known as a condensation reaction, which is the reaction that usually occurs between amino acids. Dehydration because once you react one amino acid to another, the result will be one water molecule. Again, if you react two amino acids, you will get one water molecule as the byproduct. And what is the major product in the reaction between the two amino acids? Of course, you will have a product known as peptide. Again, in the dehydration synthesis reaction, the byproduct is water, one water molecule, and the major product is the peptide. The peptide is already a molecule which is consist of two or more amino acids. And why is it known as a dehydration synthesis? Because again, out of the reaction between the two amino acids, we could have a byproduct, which is a water molecule. Now, the peptide bond is also known as an amide bond. Why? Because the resulting group, C double bond O and H, is known as your amide group. Remember in my previous video, C double bond O and H is an amide group, right? So this specific group is formed by combining two amino acids. This one, again, is known as an amide bond, or amide, can also be called as a peptide bond. Right, you have there, the resulting molecule is an amide. So you have there C double bond O and H. That is the molecular formula for an amide group. The four-atom functional group C double bond O and H is called a peptide link. Polypeptides and proteins are chains of amino acids held together by peptide bonds, as is the backbone of proteins. So as I've mentioned, the protein, or sorry, a specific protein is composed of a long chain of amino acid. So there will be, of course, a lot of amino acids that need to be linked together to form such specific protein. The peptide bonds covalently join the alpha carboxyl group of each amino acid to the alpha amino group of the next amino acid in the protein chain. And that is exactly what I was talking about in the previous slide, right? Remember, you have here one amino acid and another amino acid here. And the way that they are reacted to each other involves the removal of OH from the carboxyl group of the first amino acid and the removal of one H from the amino group of the second amino acid, forming now water. So we remove the OH here and we remove the H here. So we remove the OH, we have the remaining C double bond O, and then the remaining NH here in the second amino acid. So the OH was released, the H here was released, and the released OH and H form again water. Now the remaining C double bond O and H will be your amide or peptide link or peptide bond. So this is the clearer version of the explanation when peptide bond is being formed. So again, you have one amino acid here, and this is the other amino acid. And during the dehydration process or the condensation reaction, the OH here in the first amino acid will be released, and the H here from the amino group of the second amino acid will also be released, forming now water. Now the remaining molecules, C double bond O and the NH, will need to be linked together, forming your amide. Sorry for the background noise. But anyway, this is the amide. So once linked, this is the final product. And the final product is already known as a peptide because again, it is composed of two or more amino acids. Again, this is now a peptide because it is composed of more than one amino acid. So you have here two amino acids linked together. And this specific group formed here is known as your amide group or peptide group. Other term, peptide bond, amide bond, or you can also, substitute the name bond by the word link. So you have peptide link or amide link. Right? So you have a couple of, terminologies that you can use to call this specific structure. Now, these are just some of the characteristics of a peptide bond. The atoms involved in the peptide bond form a rigid planar unit. Because of its partial double bond character, which is contributed by the C double bond O coming from the first amino acid, the peptide bond has no freedom of rotation. However, the bonds involving the alpha carbon can rotate freely. As what was mentioned, the peptide bond has that specific, rigid structure, which is contributed by the double bond character. From the C double bond O and H molecular structure. So the presence of the double bond here makes this specific part of the molecule rigid, which means there is no rotation at all, which means there will be no movement in this specific area. Again, no movement in this specific area, no rotation. Peptide bonds are extremely stable, which means it would really require a high amount of energy in order for the bonds to break. Cleavage of the peptide bonds generally involves the action of the proteolytic enzymes. So please do remember, guys, again, that peptide bonds are really strong. They are covalent bonds that need to have a high amount of energy in order for them to break. But since we need to continuously break down molecules in the body, especially proteins, again, since we need to continuously break down molecules in the body, especially proteins, we do really need to have that specific process wherein proteins are easily cleaved or broken. Now, since it would be a waste of too much energy if we just rely on the energy alone to break the proteins. So we resort to using proteolytic enzymes. Proteolytic enzymes are types of enzymes that basically target the peptide bond. So what the proteolytic enzymes do is that they will act on the peptide bonds. If this is one amino acid and another amino acid, this is the bond that was made between the two amino acids. The proteolytic enzyme will have to act upon the peptide bond and break it in order to release two amino acids. So in this way, the protein is gradually being degraded or broken down. All right, now let's have the peptide. So amino acids bond together to form an unbranched peptide. Right? So again, as I've mentioned, a peptide is two or more amino acids that are bound together or that are linked together through the peptide bonds or through amide bonds. Dipeptide, if there are only two amino acid residues, or that means there are only two amino acids that are linked together. Oligopeptide, on the other hand, would be a peptide with 10 to 20 amino acid residues. Polypeptide, on the other hand, is already a long and branched chain of amino acids. Okay, now let's have the writing of structural formula and abbreviated formula of your peptides. So in order for us to somehow have that specific capability to name a peptide, we first need to somehow remember the abbreviation of or for the names of the amino acids, because again, peptides are formed by combining amino acids. And before we name a peptide, we first need to identify the specific amino acids involved or contained in that specific peptide. So again, you got to remember the 3-letter and 1-letter name abbreviation of your amino acids. So you have to remember, what does ASN stand for? What does LEU stand for? Because yeah, that will be required of you so that you will be able to know how to name a specific peptide. Now we have to learn how to name your peptide. So naming your peptide, we have to follow some rules. Rule number 1, the C-terminal amino acid residues keeps its full amino acid name. First, let's identify what is your C-terminal amino acid. Remember that a peptide is composed of several amino acids that are bound together through peptide bonds. Now, which of the 3 amino acids here is known as the C-terminal amino acid? Very easy lang. All we have to do is to locate the amino acid that still has the carboxyl group in its structure. So the only amino acid that still has the carboxyl group in its structure is the amino acid that is located in the far right or the far right end of the amino acid chain. Again, the C-terminal amino acid is the amino acid that basically has or still has that specific carboxyl group in its structure. In this case, the C-terminal amino acid is the amino acid that we can locate in the far right end or pinakapunta sa tuo or pinakapunta sa right end or punta sang imo nga amino acid. So in this case, this is your C-terminal amino acid. If this specific amino acid is your methionine, that means we can expect that the name of the peptide also has the term methionine in it. Again, if the name of the peptide, if the name of the C-terminal amino acid is methionine, we can expect that the name of the peptide will have the term methionine in it. Guys, take note, this is the C-terminal amino acid. How about ang sa piyak nga pinakapuntang amino acid? This one naman is known as your N-terminal amino acid. Again, this is known as your N-terminal amino acid. Miss, why is it called as the N-terminal amino acid? Because it is the only amino acid that still has the complete NH2 or amino group in its structure. So, meron tayo NH2. Remember, may free pa siya nga NH2 sa iyang structure. Sorry. NH2. Right. Sa punta, dere, may ara ka C double bond OH. Dere naman sa wala, may ara ka naman NH2, amino group. Right? This is known as your N-terminal amino acid. This is your C-terminal amino acid. Now, all other amino acid residues have names that end in YL or il. The YL suffix replaces the INE or in or ic acid ending of the amino acid name. So we only have some exceptions in this rule for the tryptophan, cysteine, glutamine, and asparagine. So for example, let's go back. If we have, let's start with the one that is easiest. So if we have two amino acids, this is known as your dipeptide. Again, two amino acids bound together via the peptide bond. That peptide that is formed is known as your dipeptide. Di, which means two, right? Now, this one is already given as the C-terminal amino acid. Example, the name of this amino acid is methionine. Again, the name of this amino acid is methionine. Now, how would you name this one? Example, this one has the name serine. Again, example, this amino acid is serine. Now, the first rule is again, the C-terminal amino acid residue keeps its full name. So given that, we will be able to write the full name methionine in the peptide name. How about the serine? So for the serine, we are going to modify that specific term. Instead of serine, we need to write serine. Again, instead of serine, we need to write serine because we have to change the INE. Again, we have to change the INE into YL. So SERYL, serine. Right? Serine. So the first term that we will put in the name of our peptide is serine because the name of the first amino acid is serine. And the second name is methionine. Why methionine, miss? because the last amino acid here is the amino acid methionine, and it is the C-terminal amino acid. Therefore, it has to keep its full amino acid name without any modification. For the tryptophan, all we have to do is just remove the AN and attach it with YL. So we will have the term tryptophil. For cysteine, cysteinyl. For glutamine, glutaminyl. And asparagine, asparaginyl. So if we have an amino acid here, two amino acids here that are bound by peptide bonds. Again, this is termed as dipeptide. Now, example, the name of this amino acid is glutamine. And the name of the second amino acid is, let's say, alanine. So can you guess the name or the final name of the peptide? So the final name of the peptide is glutaminyl. And we just have to copy this name, alanine. And yes, we have to write it in a way nga mangin one word lang siya. Alright, so again guys, this is just a one word. When we name peptides, we only, when we give the full name of the peptide, we are just writing the name in one word. So regardless how many amino acids are there in the amino acid chain, all we have to do lang is to attach all of those names to form one word or one name for that peptide. So for the words glutaminyl and alanine, there is no space at all. One word lang siya. And rule number 3, the amino acid naming sequence begins at the N-terminal amino acid residue. So I've already taught you how to identify which amino acid is the C-terminal amino acid and which one is the N-terminal amino acid. So hapos lang no, whoever or whichever amino acid that is found on the far left or sa pinakapunta sa left sang imong amino acid chain, siya na si N-terminal or N-terminus. For example, this is your amino acid chain. Kung sino ang amino acid or whichever amino acid that is found on the far right end of your amino acid chain, that is your C-terminal amino acid or the C-terminus. So we name the peptide starting from the N-terminal until the C-terminal. Now let's have an example. We will assign IUPAC names to these two amino acid sequence or two peptides. So the first one, let's first identify what specific type of peptide we have here. To identify what kind of peptide we have, let's first count how many amino acids there are in the amino acid sequence. So we have the glutamic acid, that's 1. We have serine, that's 2. And we have alanine, that's 3. And so the type of peptide that we have is tripeptide because we have 3 amino acids here in the amino acid sequence. How about the number 2? The number 2 has how many amino acids? 1 for glycine, tyrosine, 2, leucine, 3, and valine, 4. So it has 4 amino acids. That means what we have here is a tetrapeptide. Alright, next, let's go now in naming this specific peptide. So the first one is glutamic acid. Remember, the manner of naming the peptide is from N terminus to C terminus, or N terminal to C terminal, which means we will first start naming from left until right. So for number 1, let's have your glutamic acid. Your glutamic acid, the start should be capital letter G. Right. From glutamic acid, we will only have glutamyl. Again, from glutamic acid, we will only have glutamyl because we need to change the IC acid into YL. Glutamyl next. For the second amino acid, we have serine. And if we put YL there, we will have seryl. And again, there has to be no space at all. Again, no space at all between two terms. Right, so we're down to the last amino acid, which is alanine. Remember that whatever amino acid that is found in the C terminal, they tend to retain their name. So alanine still be written as alanine. So the name of our tripeptide now is glutamyl seryl alanine. Now if we would like to be very strict, we can actually name this as L, glutamyl, L, seryl, alanine. So why do we put L here? Remember that your amino acids are usually in their L form. Again, most of your amino acids are in their L forms. But since it's already given na laban or most of your amino acids are in L form, that's why sometimes we just omit the L indication in the name of the peptide. So that's why don't be shocked if we will just require you to write the term without the L. Right? Hindi nila pabutangan L because given naman nga most amino acids are in their L form. So all you have to do is just write glutamyl seryl alanine. No need to put the L somewhere in the name. Next, we have the tetrapeptide, glycine. Let's start with the glycine. So glycine will have the glycyl, of course, glycyl. Next, tyrosine, tyrosyl. Next, leucine. Of course, that's leucyl. And lastly, we have valine. We'll just copy the name of the amino acid in the C-terminal area. So we have glycyl, tyrosyl, lysyl, valine. Now that's the name of your tetrapeptide.